most microscopic things when the microscope was discovered, when people started looking at them. And these were a lot of usually wealthy doctors who took up microscopy as a hobby. So a lot of our early scientists were doctors who did phycology, mycology, any kind of microscopy with animals or that as a hobby. So these here are pictures of live diatoms, and most all of these are from Pennicamp. These are from BioBlitz, and I separate them into planktonic and benthic forms. The planktonic forms are most often have a radial organization like this one here on the bottom, like a circle. And if you turn that circle, think of it like a coin, turn that coin on its side and put a whole bunch together, you can form a chain, and that's what you have up above. And then what makes a benthic diatom unique and what really caused the evolution of these things to expand, pardon me, was a development of what they call a rafe. A rafe is a slit in the diatom skeleton, and we can see that here on the very upper right-hand specimen there, and that's that little kind of curved shape through there, and that is how they move. So they actually secrete some mucilage through there, and like a tractor, these things move around. And when that feature evolved in diatoms, that allowed them to occupy a whole bunch of more habitats, and the amount of the species diversity exploded about 50, 60 million years ago. And you see that bright golden color. One other thing to notice too is also up in this where it says the benthic, you maybe see some little spheres that appear like blue. Those are oil droplets. So they actually produce oil, and that's part of their storage product. And it's actually believed that diatoms are the source of a lot of the major petroleum deposits in the world. But probably the most important thing that we should remember about diatoms is that they fix carbon as part of photosynthesis, and as a byproduct, they produce oxygen. And the amount of oxygen they produce corresponds to about every fourth breath that we take was oxygen that was split from water by a diatom. So every fourth breath, you can thank a diatom for that. And those other quarter, 50%, you already know about the Amazon rainforest and the big lungs of the world. Well, these planktonic diatoms out in the middle of the ocean, they also are big supporters of that. They're also used to infer environmental history. They go back and they preserve very well in the sediments, and they have ecological niches dependent upon the environmental conditions. So we can go back, look in sediment cores, and infer what the history was of a specific area through sediment cores and through diatoms. So that's the crash course in diatoms, and right now you're pretty much all experts. You qualify as experts in Key Largo, at least. This describes that journey of discovery, and a lot of it is a bunch of serendipitous events. There's a lot of life lessons in here with hubris, sharing, patience. I said it all started with a simple phone call. That woman on top is Dr. Nicole Stacy, and she's a veterinary pathologist at University of Florida. And she called me on the phone and said, "Hey, we got a problem. Problem is, I think the veterinary pathologists are misidentifying what I believe are diatoms, and they're recording them as harmful parasite eggs, when in fact they're probably not harmful at all." So what she had done, she had sent some stuff to me, and that led to 2 decades of work and collaborations across the world, and a whole network of people, including we have people from the Netherlands, and then even in China. So this was the problem that Nicole was talking about. The picture on the left-hand side is a picture which appeared in one of the veterinary journals, and the caption under that picture was, "This is an egg suggestive of protozoan origin." Now, I obviously wouldn't expect many people to know what that thing is, but if you were a possibly, if you were a diatomus, you might be able to recognize it. I recognized it as a marine diatom. And what we see here, there's a mucilage bubble that's on the outside, and then in the middle that there's this diatom valve, which is the one on the right. And you see two plastids, north and south, and you see a little crossbar, which you can also see in this scanning electron micrograph over here. So this was truly a problem for the veterinary pathologists. So these are the pictures he sent me. He sent me about 30 pictures. They're not great pictures. They all have this purple stain on it. This is something that the pathologists use, and they examine all types of body fluids and skin smears. And what we see in this one, those are some skin cells or some cells inside the manatee teat fluid, and that little hazy area up front is a diatom. You really, I couldn't tell exactly what it is, but I could say, yeah, that I can tell that's a diatom. Now, diatoms have two valves that look like a box, okay? And you might be able to make out here like two parts of a box with little bands in between. So I can recognize this as a diatom. And at the end of looking at her 30 slides or pictures, I could say, okay, yeah, these are diatoms. These are not harmful parasite eggs. They're diatoms. Great, end of story. We're done. But I asked her. I said, hey, these things are pretty interesting. I said, do you happen to have any, raw material that I could prepare and make permanent slides at and look at under the microscope or electron microscope? And she said, oh, I don't have that. This is all I got. So okay, end the story, right? This is where like a little serendipity comes in, and Hillary, this is where you come in too, in sort of ways. So about 2 weeks later, I just by chance, I was sitting at my desk, and my desk is right here at the Everglades Ranger Station. And one of the park rangers knocks on my door and says, "Hey Tom, we got a dead manatee that's clogging up one of the cuts in the bay. Can you come on out and help us move it out of the way?" I think rotten manatee. Most people would say, I got something else going on, but I'll pass. But I said, hold on a minute. Let me grab my toothbrush and I'll be right there. And the reason I say toothbrush is because that's what I use to sample these guys. We use a little toothbrush to brush these off of these animals. So this was a picture of one of the dead manatees out in there. And this is a picture of what the skin looks like. After the manatee dies, the skin separates. It looks like a piece of leather or a piece of old shirt. So I actually brought that back in the lab, and I could soak that in acid and get all the diatoms off there, and I actually see what she had sent me in much better. And what I had found, well, I guess I should bring apart this part about hubris, because when Nicole first asked me to look at these things, I told her, oh yeah, these diatoms are going to be no problem, because they're going to be the same diatoms that grow on the benthos and on the bottom in the sediment, and I know all them, so I'll be able to identify them. I'll tell you what they are. So when I finally got to look at these on the manatee, I noticed a few things. One, diversity was low. There wasn't a whole lot of variety compared to if we look at the a diatom sample, let's say from the sediments out here in Pennicamp, we may see 100 different species on a slide. Very, very diverse. This one only had about a dozen, and of that dozen, it was really only dominated by 3 species, and those 3 species were about 90% of these species. But one thing I was to my chagrin was I looked at these and I said, I have no idea even what genus these things belong to, not let alone species. And I figured I had 100 books, so I got a couple of file cabinets full of the primary literature. I couldn't identify these things at all. So at that point, sorry. At that point, I contacted a friend of mine, Mike Sullivan, and Mike was the editor of Diatom Research Journal for like 20 years. And I said, Mike, I got something. I'm embarrassed to tell you, but I can't even identify what these things are. I sent him a picture, and he replied back in about a day, pretty quickly. And I could tell he was like laughing in his email. And he said, yeah. He said, yeah, what you got there? He said, those belong to a genus which has only been found on whales. And there's only, maybe a handful of papers on it. So it's very rare literature. But he was the editor. He knew where it existed. And we checked that out. And right away I could figure, okay, yes, these diatoms we're seeing on the manatee belong to this genus, but they're none of the species that occur on whales. They're all brand new undescribed species. But I said these were these whale diatoms. So this is a picture of two orcas. I don't remember if these are Antarctic orcas or whether they're up in the Arctic, but that golden film on the bottom of them, those are diatoms. And believe it or not, these things were known 100 years ago and were actually one of the first diatom indicators to be used by industry. The whalers would specifically notice the whales that had the yellow diatoms on. They didn't know they were diatoms, but they called them sulfur bottom whales. And these sulfur bottom whales, specifically the blue whales, were the ones to target for killing, because those whales had larger amounts of blubber and oil in them. So those were the ones that they specifically selected. They selected the ones with the golden bottom. So because of that led to some further attention and further study of what is that golden bottom on them, and it was later discovered that these were these three species on top. This one on the bottom is actually from 2012, described from an Amazonian turtle, not a sea turtle, a freshwater turtle, but interestingly, this one colonial nests on beaches, just like sea turtles. So we don't know the significance of that, but we find these things on sea turtles also. So after that, okay, we said, great, we've got these neat diatoms, but we've only been able to look at them dead. We'd like to see what they look like alive. Okay, we'd like to describe, their plastid, their chlorophyll. We'd also like to grow them in culture and extract their DNA and figure out how they're related to other diatoms. So we got an opportunity to sample manatees during the annual health assessments that occur in Crystal River, where they actually gather them up. And this is one of the patients coming in for his annual exam, and they'll draw blood, they'll weigh them, measure them, and get a whole bunch of samples. And so I was able to sample some alive material. So what does the live material look like? This on the manatee, that's a little, obviously a close up of the manatee skin. And just those little smudgy, slimy areas, and those are loaded with diatom. Hundreds of diatoms, hundreds of these tertiary-colored diatoms, all in that slime. They're all over the skin of these manatees. So we said we had two main goals. We wanted to get them to culture, okay, and describe what they look like, what the plastids look like. We failed on both accounts. We're unable to describe the plastids, and these little isolations of these cells did not survive in culture. But we observed something much more exciting. The reason we were unable to describe the plastids and photosynthesis is because they don't photosynthesize and they don't have chlorophyll. So we mentioned 25,000 diatoms. Out of 25,000 diatoms before this study, there were only 7 that were known to be heterotrophic that lacked plastids. All 4 Tertiary species, diatom species that we observed on the manatee were what they call apochlorotic, lacking chlorophyll, lacking color, and were heterotrophic. So these guys behaved like animals. They weren't fixing energy from the sun. They were getting their energy for some compounds, some mystery, would still remain a mystery, something from the bacterial film or the skin of these manatees. So this is very exciting because now we know that Diatoms have lost the ability of photosynthesis at least twice during their evolution. And this obviously is very, very rare. So out of what 25,000 plus, we now have 10. Okay. So something very interesting and neat is going on on the manatees. Oh, at the same time, from those Crystal River manatees, we described, we also found 3 additional new of this very specialized genus. And I said this genus is only found on manatees, sea turtles, and whales. It's not found in the surrounding environment. It's not found in the plankton. It's unique to these animals. So that also means that these animals are endangered. Everything that's unique and living on them is also just as endangered as their host. And in the case of these guys, who obviously have a very unique metabolism, we lose that. We lose a very unique biochemical pathways that we know very little about just yet. So I'll show you just a little picture, some more pictures of what they look like. This is what they look like alive. They're kind of clear. You see the oil droplets. There's a lot of oil in them. This one here has this strange like ridge that comes up over the top of it. That is very unique, not seen in any diatom sea. And what happens when something comes into a brand new area and then it evolves in this brand new isolated area? It lacks competition. So there's a lot of things that can happen during evolution. It can experiment with a whole lot of different things that might be weeded out with a lot of competition, but might exist here. So we see some strange things. Another species that has these, little what I call shark fin-like costae. So appears kind of fuzzy. And at the very end of writing that up, writing up that report, one of the final things was describing the habitat that these diatoms live in. Well, okay, obviously that we described they live on the skin of manatees. Well, what else is there? And we asked about, what was that? What was that red felt? The felt is a red algae. Okay, so all right, well, we'll take a crack at identifying what that red algae is, and thinking again, still having this thought in my head and everyone else's head that the stuff that grows on these animals is no different than the stuff that grows on the sediment, on the walls, rocks. So we were thinking it was going to be just one of the existing species of red algae belonging to this genus called Polysiphonia. Well, we did a screening of it. We got DNA and didn't match anything. So we had another brand new life form that only occurs on these manatees. And that's this red algae. We look at this red algae in terms of its morphology, and it's also very unique within the genus. So the genus has about 100 species in it. And most species of that red algae either attached to their substrate with something called a holdfast, which is, you think of like your hand, like a thumb, like this, like a pad, holds onto it like a pad. Most other species will form like a mat of horizontal rhizomes that spread out like crabgrass, okay, like that. This one was different. This one had sort of like a taproot. It grew vertically, straight down. And we deduced the reason for that is the manatees shed their skin. And as the manatees shed the skin, if you shed it all the time, you would lose the algae would lose its contact and it wouldn't survive, it would disappear. So by having a vertical taproot that goes through multiple layers of skin, which it did, it can persist and be especially adapted to the manatee skin environment. Just show you quickly we looked at the sea turtles. Part of the stuff that Nicole sent was also samples from sea turtles. We noticed the same thing. We got, live samples and good samples from sea turtles. And we found these guys over all 7 species of sea turtle all the way to Australia. And again, new genera, new species, again, only on these animals, not in the surrounding environment. Specialized taxa, the community, again, low diversity, but dominated by these very, very specialized species. So really, one of the questions that remained when we were doing this was, okay, well, does this stuff really differ from anything else that's out there floating in the ocean? What if we just looked at the plastic debris and flotsam? So there was this group of Zeri atoll, and they worked in the Persian Gulf, and they collected a whole bunch of flotsam. That's all the stuff that floats, just like sea turtles would. Okay, and they looked at the composition of the diatoms on them and compared that to the composition of the sea turtle that was on them. I believe those are green sea turtles over there. And they found 37 species. Only 4 of those 37 species was actually on both substrates. And those 4 species were common benthic taxa that occurred on the sediments. So these were taxa we know. These weren't specialized taxa. But on the sea turtle, what comprised the most were two specialized diatoms in dominance. Again, a specialized flora just on the sea turtle carapace. I guess I should mention briefly, we also, when we looked at sea turtles, we found that there was a different composition, different species that occurred on the carapace and different species that occurred on the skin. So 2 different environments there, also with different specialized communities. This here is a cladogram. This is like a tree. Okay, this shows the relationship among these species, and you can either produce this tree using DNA similarities, or in this case, this was through morphological analyses. Very, very detailed. I'm not going to get into it, but what I'd like you to get here is that the sea turtles, the cetaceans, the whales, freshwater turtle, and the manatees, they all had related communities, but distinctly different. And these communities were unshared. So there was a specific community, specific to manatees, specific to cetaceans, specific to sea turtles. But seems to be shared amongst the, let's say, sea turtle species, loggerheads, greens, olive ridleys. They'd share very similar, the same species, and similar thing with the cetaceans. So question that we ask and we'd like to do, part of our studies now, is, okay, we have these different specialized communities living on these animals. How did they get here? Where did they originate from? Okay, so did they, was it one colonization and then somehow it spread from a manatee to a sea turtle to a whale? Was it three separate ones by the same diatom? All of that we're currently studying and trying to figure out, but we still don't know. But we can use some ecological principles to guide us in our investigation. One of those is called island biogeography. So many of you may be familiar how if you go to some of the isolated islands out in the oceans, they have a unique flora. You probably all know about the Galapagos and their unique flora out there because they're isolated. So the flora and fauna that are in the Galapagos originated from original colonists that got on there, and they evolved into different species because they were isolated, geographically isolated. Now, this also occurs on mountaintops, something that lives only up in the very cold areas of mountaintops. There's a big separation between the lowlands and the next mountaintop. So you also have what they call an endemic flora, a local specialized flora that live on these mountaintops, same as isolated islands. And we think of whales and sea turtles as being these islands of isolation out in the middle of the big wide ocean, an island of hard substrate that doesn't exist, that doesn't exist close by. So anything that gets on them, you fall off it, you're dead. You stay on it and survive. You're not going to interact with anything else, and things are going to evolve into new species. Another concept is something called adaptive radiation. This is where your birds come into play. Okay, any of you are familiar with Darwin's finches. So going back to the Galapagos, one of the birds that colonized the Galapagos early on evolved into a new species, and that species went in 3 different directions, and that single species evolved into like 15 different species, because the Galapagos there were a lot of ecological niches, little areas that roles were unfilled, and that bird evolved to occupy these niches, occupy different roles on there, and evolved into different species. We believe this is what's happening on the manatees and the whales. We had one of these diatoms come on. It evolved in terms of the manatees, 6 different species. We don't know the niche yet. We don't know what the role is. So that's part of our studies, trying to figure out, how do these guys divvy up the resource? How are they specialized actually on the skin itself? And this is just a little illustration of that. So while all that is going on, I also in 2019 I received an email from another veterinarian working out in Washington. And I don't know if any of you are familiar with that. They've been having a tough time with the gray whales. A lot of gray whales are dying. They have a big mortality event over the last few years. They're emaciated. And a lot of them are being found washed up on the shores. Trying to figure out what's going on. One thing that Jessie Huggins, she's the veterinarian out there. One thing she had noticed on some of these whales was that the baleen was a strange color. So the baleen is what some of the whales that have an organ baleen to either filter plankton, or in the terms of these gray whales, they actually feed on the bottom benthos. They dig a whole bunch of sediment up and they filter this through their baleen, which is like a comb, okay, and then they swallow everything on the comb. Well, she had noticed that some of these were the baleen was red color and when it should be gray. So she asked, "Hey, could you take a look at what's on the baleen and tell us what you see?" And this is where again another hubris comes in. I said, "Well, yeah, I'll be happy to take a look at that, but I'm really interested what's on the skin. I'm not really interested what's on there, but I'm happy to take a look and find that for you." But so she started sending me skin samples and samples from the baleen. And you want to guess what I saw, what the results were there? Well, I didn't find much of anything on the skin other than incidental plankton diatoms and stuff from the beach. I didn't find any specialized diatoms in the skin. But on the baleen, Turisticola showed up again. A new species, but again, that same group. And you look back and you go, "What? Why is my thinking wrong there?" Well, you know what? That little filter you're talking about is filtering out stuff that's is a few inches big. Krill's pretty big. It's a shrimp. The stuff that grows on these diatoms are orders of magnitude smaller. So they can stick on there regardless of what gets flushed through them. So this is a little just showing this what it looks like. This one was unique. It had a totally different structures. But what's common to this genus is this specialized structure that they call the butterfly structure. That is a close-up of it on the left-hand side. We don't know exactly what that's for. It's a thickening of the glass of the silica, like sort of buttresses. And if you think about, God, what could that be for? Well, maybe this has got something to do with the way these whales dive down to thousands of feet, the changes in pressure. So maybe these diatoms need a little extra strengthening, and this butterfly structure may be part of that. So that's another part of our investigations. So again, what have we learned? We learned that okay, there truly is specialized flora known as epizoic taxa. Before 2013, I thought the stuff that was living on these animals was the same stuff again you would find growing on the floor, on the ocean floor, but it's not. It's a specialized, unique flora, and that flora is just as endangered as its hosts. What's going on these animals is the same process, ecological process that we see happening on these isolated islands. So we see adaptive radiation. We see those common principles. And so there's a lot more epizoic algal discovery waiting discovery. I'm still receiving samples. I most recently got a sample this fall from bottlenose dolphins, wild bottlenose dolphins from the East Coast, and those had some of those known whale diatoms, but also a new one. So there's still a lot of stuff out there to be described on their skin. Yes, on their skin. Yeah, yeah. And she actually had some nice pictures, nice pictures of it. So this is part of the stranding network. Um, so with that, I'd like to end our talk and open this up to questions. And thank all the animal capture teams. Obviously, these samples are very rare and very difficult to come by. How do you get a sample from a whale? Right? So you can see a lot of our samples from whales are stuff that's already dead. We're still actively trying to get live material so we can get DNA from them and hopefully grow them. But again, how are you going to get a live sample from a blue whale? We sort of got to get lucky with some strandings and get some fresh material. Um, yeah. Okay. Thank you. Yes.
So I'm interested in the intricate silica formations that vary from diatom to diatom. How have you figured out how that comes about? Do they understand reproductive?
Yeah, they have something. They have a little process called the silica deposition vesicle, and inside this little vesicle, there is sort of a template that is sort of formed, and they lay down this template, and it starts out from the center of the diatom, and then builds out from the center. Um, yeah, there was a whole set of scientists and literature on that, and obviously for nanotechnology, they're very interested in that. They would love to be able to know how do diatoms produce these structures, because they would like to build these structures in on their silicon wafers and stuff like that. So it's something that nature does that maybe better than we do, that we still don't know. Yes.
Between freshwater and saltwater manatees?
Repeat the question.
Yeah, I'm sorry. Yes, I'll repeat that question. Yes, sorry. The question was there a difference between the type or character of the silica between freshwater diatoms and saltwater diatoms? No, but there are differences depending upon the silicate concentration in the water. So, if you go out in the open ocean, well, first of all, you figure the source of silicate. Source of silicate is from silicate rocks which erode and eventually come into the streams, and then that silicate washes out into the coastal waters in the ocean. You go out to the middle of the ocean, there's very, very little silicate. So you'll notice that the diatoms out there have very, very, very thin frustules. But in areas where you may have a lot of silica, they'll be very robust, very, very thick. And those stuff obviously is the stuff you'll find actually in sediment cores being preserved for millions of years. The really thick, heavy stuff. Yes.
How does the diatoms move from animal to animal, like a turtle from an egg?
Yeah.
Get it.
That's an excellent question. So if these things are specialized, I'm sorry, I need to repeat that question. The question was, how do these diatoms persist on these animals? How do they get from one generation to the next generation? And Barbara mentioned the problem of the egg, the sea turtle egg. Sea turtle egg is sterile. Okay, mom lays it, mom gone. Okay, so we know from manatees and whales, they're live bearers. So the flora gets transferred from mom to calf. Persistent. Sea turtles, sterile egg. What happens? There should be none of these diatoms on juvenile sea turtles. So we look at juvenile sea turtles, and we haven't found them. We have found them, later on, older ones. In fact, that we presented at a meeting, and I alarmed the sea turtle crowd when I told them that their sea turtles were loaded with STDs, and they said, and they never. So not those type of STDs. I'm talking about sexually transmitted diatoms. Because when the sea turtles got together to mate, that's when they would contact each other and again inoculate each other with the flora. Yes.
With the sea turtle, you said that there's different species on the neck versus the shell. Are they, I guess, fastening to each structure with the same anchors? Are they like within partitioning, or some How soon is it on?
Yeah, so you see, the question was, and how do these diatoms attach to the sea turtles? What's the difference between attaching to the carapace and versus that of the skin? There've been a few studies. My colleague Roxana Majeska, who's from Poland and now in Norway, but she's going to be coming here to the Keys in a few weeks and spending time with us. But she has specifically gathered intact. You may have seen that picture with the scalpel. She was getting a little piece of the carapace from the scalpel. She'll take that whole piece, coat it, and then put it under the electron microscope and actually see how things are attached. And a lot of these diatoms, when they're attached, they're in the upright position, and they'll have a basal portion, and that basal portion will sometimes be embedded into the top layer of the skin, but oftentimes it just appears to be embedded in a little layer of mucilage. So that's on the skin. On the carapace, you mentioned this stalk of these guys living on a stalk, and the stalk was mucilage, and that attached with a pad. Just again, like your hand with a pad. So there are different attachments. Again, that'd be great. Looking more at these things in situ, we might be able to tell, what are these niches? How do these divide up? Why are there so many different species? Yes. Yes, Joanne.
Could you conclude that if there is more diatoms on the baleen instead of on the skin, that it's already getting sick, or are you not that far?
Well, that's something. So the question. I'm going to try to repeat that question. The question was, can you basically tell if the amount of diatoms or the composition of diatoms is an indicator of host health? Are these things detrimental? Are they do they come in maybe feeding on dead skin? We just started to look at that with we have a data set from harbor porpoises from the stranding network, and part of the stranding network they collect data of the appearance, the health of the host animal. And we just ran a bunch of statistics. I mean, this is just last week. I just ran the statistics last week on that, and we found that on that available data, we did not find any relationship. So it's not that none exists. It just means that we possibly don't have the right data. We're not looking in the right place yet. So again, we'll look at these things, and we're going to refine our methods and hope we tell. Yes.
How do they multiply?
Sure, that's a great question. How do they multiply? Okay, so these single-celled algae, they mostly multiply by division. So one cell it's like a box, right? The box splits open, and then a new side to this box forms, and a new side to this box forms. And what's interesting about these diatoms is the new form forms inside the old form. So as you look at a population over time that you have in a culture, the size of the diatom would get repeatedly get smaller and smaller, smaller, and then it reaches a critical minimum size length, and then sex occurs. It produces gametes, and then releases these gametes. The gametes will go through meiosis. The cells form a zygote, and they form a brand new diatom at the very top end of the size range. And they start that process all over again. They divide, get smaller, reaches critical size, go through sex, regenerate the original maximum size.
I think this is the most interesting talk I've ever listened to.
I am, thank you. I'm glad that $100 paid off. Thank you. Yes, Chris.
The feeding situation on the whales, did the diatoms affect the whales feeding? That's why they were. What was the conclusion?
Well, okay, the whales seem to be emaciated, and the current theory is that the waters have warmed, like the whole changing oceans we have, and the prey has shifted, and the whales, I guess, haven't quite changed their habits, and they are not getting enough food. So that's a theory. It's not proven, but that's about where they're at right now. Sorry.
But it kind of suggested that diatoms had affected their babies, or the reason why diatoms showed up in the babies because they were beasts?
Well, the thing is, nobody looked before. Well, it's like, I can't say that yet. There's been very few observations of that. So these diatoms that occur in the baleen probably occur natural, just like we have mites living on our eyelids. This is something that evolved on them, that is just a natural community that lives on them, that lives on that ecosystem. But it's interesting you mentioned that, because we go through the literature. I mentioned the last 100 years, there's been only about 20 papers on whale diatoms, but it was important early on. And when they were whaling, they're both here and throughout the world, and later on continued in Japan. That's where the papers were produced. And you go back to a paper from the 1930s, and there's a brief, tiny little paragraph in there about one observation of baleen, and they mentioned two things that looked. One looked like a navicula, one that looked like this something that they called cimbella. Well, that cimbella looks like one of the ones that we just described, and that navicula looks like the other one. And so I think we had a rediscovery of what something was described, almost 100 years ago, but just sort of in passing. And what they were looking for, and what they had really noted, was they also had baleen that had that red coating on it. And at that time, they found an organism called hematophagus. Heme is blood, blood is red. And I think that was a protist. And surprisingly, I mentioned that to some of the veterinarians, and they weren't aware of hematophagus or anything. So nothing seemed to have been published on that organism since then. I didn't find it, but I suspect that's what it may be. But again, digging up old literature from the 1930s and stuff like that can be quite challenging. We search in museums, and we get out for interlibrary loans, and sometimes we get stuff that's a photocopy of a waterlogged paper. But so it's always interesting when we can consult some of those old material and use it. We always try to go back to some of those original descriptions, and we're still using descriptions from the early 1800s. Does exist. Yes.
Any more questions?
Well, thank you, Tom, very much.
You're welcome. Nothing. Thank you.
Our friends would like to present you with this gift bag.
Oh, thank you.